Hello guys and welcome back to Excel Share Academy. So yesterday we discussed like how the proteins are, like during the translation, how the SRP proteins are bringing all that complex of ribosome and mRNA and bringing towards the ER. Okay. So proceeding with that only. So now we are going to study or understand like the insertion of the proteins into the ER membrane. Okay, so as we already know, the proteins which is destined for secretion from the cell or residence within the lumen of the ER. So here we are talking about the soluble protein. Okay, in the ER, Golgi, lysosome, they are all translocated across the ER membrane, released into the lumen. Okay. However, proteins destined for incorporation, that is like you have got the plasma membrane and in that it is going to be inserted. Okay, so for the incorporation into the plasma membrane, all the membranes of these compartments are initially inserted into the ER membrane and then this gets released into the lumen. So from the ER membrane they proceed to their final destination, जो हमने last class में पढ़ा ER, Golgi, plasma membrane, or lysosomes or different organelles. So they all are going to be the soluble proteins. Okay, now integral membrane proteins. So these proteins, they will be embedded in the membrane. So like in the last lecture, I showed you like how exactly is happening this. So we have got like this, आपका translocon है यहाँ पे. Okay. So you have got the translocon. Now, when it is coming with, when it is present over here, this pushes, okay? So it will push towards this side or this side so that it gets integrated into the membrane. So ये जो आपका alpha helices रहता है, okay? So they are going to be having hydrophobic amino acids. And then here N or C terminal, here it will be your C or N terminal respectively. Okay, so in this lecture, I have got the, the figure is also there. So like for better understanding. So these are the orientation by which the proteins gets inserted into the membrane. So either you can have N terminal in the lumen, C terminal in the cytoplasm, or ulta uska, N here, C here. And you can also see that there are transmembrane proteins. Okay. So what is the difference over here? We see that in the transmembrane proteins, then the translocon has to push these many times the alpha helices. So इसमें कितने हैं? 1, 2, 3, 4, 5, 6, 7. So around 7 are there. So 7 times it is going to push into the hydrophobic region so that the protein gets inserted. Okay. I hope everyone is able to follow. Okay. Now this is that just the topology like what we have discussed yesterday. Let us just summarize what we have talked yesterday about it. So here you can see this is your ER and you have got the embedded ribosomes are there. Okay. Now this is what I was talking about. Like once these embedded ribosomes are there, so the protein goes inside of the lumen. By the time this is happening, it actually forms your vesicle. Okay. Now this vesicle goes into the Golgi where it is getting transported. Okay. Now what, this is the example of your integral protein. Okay. This is the example of integral protein. So here the protein is embedded into the ER membrane. So that entire vesicle carrying the integral protein will go and get fused with the Golgi apparatus. Okay. Now in the Golgi apparatus, it goes and gets modified according to the requirement, and slowly it will be like from the cis intermediate path and then the trans Golgi. After that, again, after the full modification is happening, again it forms the vesicle and it gets fused with the plasma membrane. So this is how the integral proteins which we have studied initially in the plasma membrane, all the transporters or the channels which is present, they get inserted into the plasma membrane like this. Okay, is it clear? Did everyone follow it like what we have just discussed now? Now we'll talk about like in Golgi apparatus what kind of modification is happening. Okay, so we there is certain like phosphate linkages are there, or you can have the glycosylation happening, right? So there are two types of glycosylation happening. That is your N-linked glycosylation is there, O-linked glycosylation is there. So we'll come to these details, like what kind of modification happens in the Golgi apparatus. Whereas in the case of ER, it is the folding. Okay, it is the folding of protein which is happening. Now, for folding of the proteins, then again you need certain proteins, enzymes, chaperones. So all these are present in the ER lumen. which helps the newly synthesized polypeptide to get completely correctly folded. Okay. Now what happens if suppose a protein is misfolded? Anyone? Shruti, would you like to give the answer?
Ma'am, it will go for degradation.
Correct. Absolutely correct. Absolutely correct. So if the proteins are not properly folded, then it goes for the degradation. So for going for the degradation means we talk about the ubiquitin. Okay, so ubiquitin based, it will go to the proteasome, and there the complete unfolded or misfolded proteins are getting degraded. Very good. So every time I'm talking about this, because now in the upcoming lectures, like in future, we are going to discuss again and again, right? अब हम हमेशा बोल देते कोई भी प्रोटीन आपका है misfolded है या unfolded है which is not fit for the function, then what we do? We say that the protein is going for the degradation, but it is not that easy. Again, you have got the regulation. There are certain proteins which identify, okay, this is misfolded. They try to bring the they try to make the protein into a correct folding, but जब नहीं होता है, then they understand, okay, ये नहीं होने वाला है. Let's not get collected. जितना ज्यादा आपका अनफोल्डेड और मिसफोल्डेड प्रोटीन कलेक्ट होगा, so they will result in the formation of the protein aggregates. Okay, so they will result in the formation of the protein aggregates. Now these protein aggregates, it's just like the junk which gets collected, or the waste which gets collected in the house. Okay, so वो चीज हमें नहीं चाहिए. Why? Because eventually, see what will happen अगर so much of waste gets collected in the house, it will smell, it will get rotten, it will cause a disease. Okay, infection spread hoga. In the same way, if the cell is there and it gets collected, protein aggregation hoga, so what will happen? This will hamper the function of other proteins which are present, and eventually what will happen? The cell is not able to function properly. Okay, so that's why we don't want any protein aggregation happening inside the cell. So it's better that we send it for the degradation, where whatever the proteins are there, the amino acids will get recycled and we can reuse it in the protein synthesis. Okay. Okay. So now most proteins are inserted into the ER membrane by the SRP 661 code translation pathway. So remember, like yesterday I was telling, slowly we'll introduce the name of the translocon in which the proteins are getting translocated, okay, from the cytoplasm. The ribosomes, the mRNA, with the help of the SRP, it goes and brings to the SRP receptor, and then the newly synthesized polypeptide goes into the translocon. That translocon का नाम है यहाँ पे 661. Okay, the normal one which we always talk about. So some transmembrane proteins have normal amino terminal signal sequence which is cleared by the signal peptidase during the translocation of the polypeptide chain through the translocon. Okay, so this is what something which we have discussed yesterday also. Now the growing amino terminal portion of the polypeptide is then translocated into the ER as the translation proceeds. So what we are saying, जैसे जैसे आपका translation होगा, the polypeptide chain will get increase in the size. वो आपका धीरे धीरे it will get inside of the ER lumen. Okay. Now, translocation of the polypeptide is halted by a membrane spanning alpha helix in the middle of the protein. So what we are saying is that if suppose the translocon understands that this particular protein which is getting synthesized, the polypeptide chain has got hydrophobic amino acid stretch continuously of around 25 to 30 amino acids, right? So it understands. So what will happen? The polypeptide chain will not go inside the ER lumen. Ab kya hoga? It will go into the the channel which is present in the translocon. If you remember, so the hydrophobic amino acids gets transported to the channel which is present in the translocon. Now, once it gets completed and again your normal amino acid starts getting synthesized, then that particular portion should be present in the hydrophobic region of the plasma membrane. Okay. I'll show the figure if it's there. Yeah, here, see. So let's understand here how it is happening. Okay, so you can see that आपका राइबोसोम हुआ, एसआरपी लेकर आया, यहाँ पे रखा, एंड यू नो दैट विद द हेल्प ऑफ द जीटीपी हाइड्रोलिसिस देन द एसआरपी गेट्स रिलीज, राइट, देन इट इज recycled for bringing another ribosome mRNA complex. Okay, now what do we see here? So we saw that the signal, so these signals are what? Your signal peptide, they are actually hydrophobic, correct? If you remember from the yesterday's class, so they are hydrophobic amino acids, right? So these hydrophobic amino acids will go into the narrow channel of the translocon, where the signal peptidase recognizes the cleavage site. It makes a cut. Okay. Now this particular portion will go for your degradation, where again recycle back to the amino acid. Now what happens is that you see the polypeptide chain is there, it is getting translocated from here. While getting translated, वो आपका ER lumen में आ रहा है. The moment it translocon understands that there is the hydrophobic alpha helices amino acids are there, so it gets stopped. यहाँ से वो फिर नीचे नहीं भेजता है, ठीक है. So here it will get synthesized and alpha helical structure बनेगा. So alpha helical structure is what? Your secondary structure, right? It is a secondary structure, alpha helices and beta sheets, right? So in this case, it is the alpha helices. So it is forming alpha helices. Now the moment it understands that, okay, यहाँ पे खत्म हो गया, now I'm gonna have the hydrophilic amino acid synthesizing polypeptide chain. The translocon, can you see this portion of your translocon? This helps in the recognition of the polypeptide. Now, since this understands now this portion, the narrow channel, the hydrophobic area of the translocon is occupied by the polypeptide chain. This particular portion of the protein gives a kick, a push. So from here, it gets inserted into the plasma membrane. And this is how, with the help of the translocon, your integral membrane proteins gets inserted or incorporated into the plasma membrane. Clear? Is it clear to everyone? Rishita, Shruti, clear?
Yes, ma'am.
Cool. Now just imagine same thing. Instead of having one alpha helices, one hydrophobic alpha helices, you are going to have two. Okay. So what is happening? Again, same scenario. SRP है, वहां पे आपने कहा आ गया, signal हट गया. Okay. Now what do we see? The moment it enters, थोड़ा सा figure drag हुआ है. The moment we see that over here that it is having the alpha helices, the only difference can you see? Yeah, in the previous figure, the end terminal was this side. In this figure, the end terminal is this side. So irrespective where the end terminal is going to be present, the translocon understands and it accordingly translocates the polypeptide chain. Okay. So what do we see? N portion is present over here and it starts forming the your hydrophobic alpha helices over here. The moment it understands over there, it gets stopped and forms a loop. This loop like structure is there. And then synthesizes. Again, it understands that यहाँ पे एक और बार आपका hydrophobic amino acids are coming. Okay. So before that, it pushes and ये आपका बाहर आ जाएगा. Then again, the second part, the second hydrophobic alpha helices will come and it will be pushed into the your plasma membrane. So this process can be repeated many times till all the multiple membrane spanning regions have been completed. So we have seen that there are GPCR they are made up of 7 alpha helices, then rhodopsin हो गए जिसमें 12 होते हैं, right? So we have seen, so ऐसे ही वो repeat करते जाएंगे procedure till all the alpha helices are inserted into the plasma membrane. Okay. So once you guys get the time, you can go through what it is being written over here. It's just the same thing. Yeah. So then there are certain proteins with some proteins with transmembrane sequence at the C terminus. They are inserted into the ER membrane by alternative pathway. Now there is another pathway for the post-translation pathway. Now what is post-translation proteins? This was about the co-translation. Did you get the difference? Co-translation में क्या था? Already the proteins are getting translated and together with the translation, it is going for the translocation. Okay. Now we have got other proteins which is going to help in the post translation pathway. यानी कि protein आपका synthesize हो गया, it is present in the cytoplasm. Now these proteins needs to be translocated into the ER membrane. वो अब कैसे होगा? So for that we have got different set of proteins. Okay. So these proteins cannot be recognized by the SRP because their C-terminal transmembrane domain does not emerge from the ribosome until translation is terminated and the completed polypeptide chain is released from the ribosome. Instead, the transmembrane domains of these proteins are recognized by a different set of proteins known as TRC40 or GET3 once the translation is complete. Okay. So these 3RC40 proteins will escort the transmembrane protein to the ER membrane where it is inserted with the help of the GET1 and GET2 receptor. तो GET1 GET2 receptor क्या कर रहे हैं यहाँ पे? किसका काम कर रहे हैं? Sex61 का काम कर रहे हैं. Okay, here you have got the portion, the figure over here. What you to understand is that instead of SRP coming and recognizing the signal peptide, यहाँ पे क्या होगा? It is not the N terminal, it is the C terminal, okay, which has got the transmembrane domain sequence is present. So instead of SRP, you have got the TRC40 protein, which goes and recognizes that sequence. Now that will be brought together and it will be inserted. Instead of Sex61, you are going to have here GET1, GET2 receptor. And they are inserted into the membrane with their short C-terminal domains on the cytosolic side, okay? So this is how these kinds of proteins are getting recognized the post-translation pathway, right? And they will get inserted into the membrane. Is it clear the difference between the two methods or the two pathways? One is for your co-translation, the another one is for your post-translation. Yes?
Ma'am, can you re-explain it?
This portion about the second part?
Yes, post-translation.
Okay. So post-translation, what happens is that, see, in this case, it is already known that the C-terminal domain is meant for your transmembrane insertion. It means the entire protein will be present in the cytolic side. Okay, ये lumen side में रहेगा, soluble side में रहेगा. Now the C portion जो हमने यहाँ पे mention किया है, it is for the insertion or incorporation into the hydrophobic phospholipids में होना चाहिए आपका ये plasma membrane में, ठीक है? So for this we have got separate proteins. So instead of SRP we have TRC40, which recognizes that hydrophobic or the transmembrane sequence. This recruits these kind of proteins to the GET1, GET2 receptor. So instead of Sex61, here the translocon is known as GET1, GET2 receptor. With this help, what they do is the transmembrane region is actually present over here, which is pushed later and inserted into the plasma membrane.
All right, ma'am.
Okay. Yeah. Tanvi, is it clear to you?
Yes, ma'am.
Okay. Now, so there are different types of proteins are there. Type 1 single pass protein and type 2 single pass proteins are there. So what is this type 1 and type 2? Can anyone guess what is it? So there was this slide where I've mentioned like type 1 kya rehta hai. So it's a signal sequence followed by a long hydrophobic patch. Okay, so from the figure itself, it's very clear. So here what happens is that in this kind of insertion, here once again, yeah. So you can see again this when you show that the ribosomes are present, please do understand that it is co-translation wala, okay? So what they have shown is mRNA is there, ribosome is there, your proteins are coming. So यहाँ पे of course signal peptidase है जो आपका signal हटा देगा. Then you have got the N terminal is there. Okay, so you can see cleaved signal sequence is there. Now N terminal is there. Stop transfer anchor sequence. Now what is this stop transfer and start transfer? Just now I have mentioned you that the moment the translocon understands that it is the alpha hydrophobic alpha amino acids, alpha helices amino acids are there. Okay, that recognition is because of the start stop sequences. Okay, so I'll come to that again. So what you see is that because of those sequences, it understands and synthesizes. Okay, so you have got the start and stop sequence. यानी कि इतना ही वो alpha hydrophobic region synthesize करेगा. Rest of it is going to be a normal polypeptide chain having exposure towards the cytoplasm. So आपने क्या देखा यहाँ पे start and stop sequences आ जाता है, and then it is going to push into the plasma membrane. Rest all is synthesized and left in the cytosol. Okay, so यहाँ पे आपका N terminal हो गया, ये आपका alpha helices region हो गया, and here your C terminal is present in the cytosol. So these are the type 1 type of polypeptide or single pass protein where only one spanning alpha helices is present. Is this much is clear? I'll talk about start and stop transfer sequence again in the later sections. There it will be more clear. Okay. Now coming to the type 2. So type 2 mein kya hoga? That you have got your N terminal at the cytoplasm side. Did you see the difference? यहाँ पे N terminal आपका lumen side में है, C terminal आपका cytosol side में. ओके, यहाँ पे N terminal आपका cytoplasm side में है, C आपका lumen side में. ओके, इतना अंदर समझ में आया in the figure? I know it is not perfectly made, but did you get the point? When you
Yes,
said
ma'am.
type 1 single pass protein, N terminal is at your lumen, ER lumen. C आपका cytosol में, ठीक है? Type 2 is where you have got N in the cytosol. Okay, now see the difference. So you have got the polypeptide chain which is being synthesized. Now once it understands that your again hydrophobic alpha helices amino acid starts, so it will start getting inserted into the translocon like this. Then it recognizes that, okay, that is enough. Only now normal synthesis is required. So ये जो hydrophobic region होगा इसको हम ऐसे push कर देंगे and resol will be synthesized. The moment it is completed, the entire mRNA has been translated. So this translocon will translocate this portion into the lumen. So that's how you have got your C terminal in the lumen and N in the cytosol. Did you see the difference in the figure? Are you able to imagine like how exactly these proteins are getting inserted into the membrane? If you understand this, then only we can go ahead. Do you want me to explain again? Is it clear to you, Shruti?
Yes, ma'am. Yeah.
Okay, okay. Now coming to the hydropathy plot. Okay, so it's not only about the connexin. I hope you know what is connexin. Any idea like what is connexin? Rishita, would you like to try giving this answer?
Yeah, I mean, the passage sort of connexon and connexin, the passage. This is what you get to see in, I mean, I'm not able to explain probably, but it is a passage sort of, which allows, right?
It is what?
I mean, passage.
Okay. So do you want to say that these are the transporters which helps in the passage of the molecules?
They are not transporters. These are more stationary, but then these are passages, is what I know.
So they are actually your, see from the word connection, you should understand thoda about this, it's related to the connecting. Okay. So when we talk about cell adhesion or cell to cell interactions, there we are going to learn like if two cells are there. So these connections are the connecting links between these two. So which allows a passage from one cytoplasm of this cell to the cytoplasm of the another. Okay, so these are the connection proteins which allows the passage of this molecule from here. Usually transporters kya karte hain? From outside to inside or inside to outside. Connection is between the two cells, adjacent cells. So yeah, it is correct. So good attempt. Now why I'm taking about connection? Because it has got the hydrophobic regions, okay, in equal quantity with its hydrophilic quantity also. So hydropathy plot, it is also known as, I think, KD plot. I think it is KD plot, if I'm not wrong. Please do study about it, okay? Exam mein KD plot aaya to aisa nahi hona chahiye ki that we haven't learned about it. KD is the name of the scientist, Kylie Doodle, ऐसा कुछ तो नाम है, मुझे भी याद नहीं आ रहा है. Just please search about it. So it is also known as KD plot or the hydropathy index, which will tell you, okay, how much hydrophobic your polypeptide chain is, depending on its amino acid sequence. So usually in the research, when you carry out certain experiments and you understand that every time you purify your protein, it is getting precipitated. Precipitated means while purifying from the cell. तो आप उसको उसके क्या कहते हैं the optimal conditions से उसको हटा रहे हो, right? You're bringing outside of the cell. So what happens most of the time, the cells are most of the proteins, they get precipitated out. That is, they get misfolded. ठीक है? So if your protein is soluble, then from one soluble region to another soluble region, it shouldn't happen. Okay, so in order to understand whether the protein is hydrophobic in nature or hydrophilic in nature, if it is hydrophilic, then we say it is soluble. तो आपको purification में problem नहीं आनी चाहिए. But if you know that your protein is hydrophobic in nature, then you will understand that कि आप उसको normal तरीके से soluble नहीं बना सकते हो, and you need to add what? Tanvi, Shruti, for hydrophobic proteins, how do you purify from the given source? I have taken this already. Think. I'll remind you. So whenever you want to purify any membrane-bound proteins, do you remember what all things I told you to add which helps in the purification of membrane-bound proteins or hydrophobic proteins? Which does not change their function or alter their structure.
Salt?
No. Think. Especially for membrane-bound proteins. Salt anyway we add to make that, to maintain that osmolarity of the cell. Tanvi, क्या है आंसर? सोचो सोचो. Okay, not able to remember. That's okay. That it is okay till you write the exam. ठीक है, एग्जाम में अच्छे से याद करके लिखना है. So if it is hydrophobic proteins or membrane bound proteins, we use detergents. Now, did you remember we have talked about it, non-ionic, right?
Yeah.
Yeah,
I thought about detergents, but then I also thought about SDS, so that alters the protein structure.
हाँ, SGS नहीं, SGS is to denature the protein, but if I don't want to denature my protein, but still I want to purify, then I use non-ionic detergent. SGS is your anionic detergent. ठीक है?
Yeah.
SGS is your anionic detergent. It has got the negative charge. Okay?
Hmm.
Yeah. So
What about Triton X100?
no, sorry, which one?
Triton X100?
I was about to say that. Non-ionic means like Triton X100 or X20. Okay, so these are what are the non-ionic detergents which you use for the membrane bound or hydrophobic proteins. Okay, so that they don't precipitate out. Okay, guys, you have to remember. Okay, now let's come back to the hydropathy plot. तो कैसे होता है? So this only the amino acid sequence will tell you how much hydrophobic amino acid residues are present. ठीक है, तो इतना ये आपको आइडिया देगा like कितना आपको नॉन आयोनिक डिटर्जेंट यूज करना होगा या करना है या नहीं करना है. ठीक है, so let's see the plot. This is just one example, and most of the time what we also now during the interview we give such plot, and then we ask like इसमें कितना पोर्शन आपका हाइड्रोफोबिक एंड कितना पोर्शन आपका हाइड्रोफिलिक है. So yes, look at the graph properly. Already it is written that 0 से ऊपर जो रहता है the positive values, okay, Since it's a hydropathy plot, so positive values will mean hydrophobic nature. Negative values will mean hydrophilic nature. Is this much clear? Now, it also shows 0 से लेके 50 तक it is having hydrophobic. From 50 to 100, it has got both hydrophilic as well as hydrophobic. Then 100 to 150 here, you have got mainly hydrophilic. Then hydrophobic, hydrophilic, hydrophobic, and then mainly hydrophilic. ठीक है? So it will not tell you that the outer of the protein structure it is hydrophobic या अंदर आपका hydrophobic है. It doesn't talk about that. Obviously, we know that most of the proteins tend to keep their hydrophobic amino acid in the center, because if they are exposed to the hydrophilic solvent, or sorry, solvent nature or aqueous solution, so they'll keep hydrophilic amino acids outside. That is in general. But this plot will not tell you. This plot will tell you that how much hydrophobicity or how much hydrophilicity is present. Okay. Now looking at the figure, what can you say, like how much hydrophobic or can you calculate how many alpha helices will be present? Guesswork only. We are not sure, but sometimes for questions for numerical, they do ask. So how many guesses, like how, as a guess, how many alpha helices might be present? Any idea? Any guess?
Ma'am, 4.
Yes, correct. Simple. So already we have mentioned that so one alpha helices in the transmembrane, it is around 25 to 30 amino acids. ठीक है? यहाँ पे भी देखो ऑलमोस्ट उतना ही आ जाएगा. इफ इट इज नॉट स्टार्टिंग फ्रॉम 0, यहाँ कहीं पे कहीं से भी शुरू हो रहा, अराउंड 25 to 30 we have here. From here to here also you can see, here to here also. So at least we know that it is more than 25 to 30 amino acids and it is coming in the hydrophobic region. So we can make a guess that at least 4 are present. We are not sure, but at least 4 is present. There might be more. Okay. But from the plot, hydropathy plot, you can understand that at least 4 alpha helices might be present. Did everyone get this point? Like how to calculate?
Yes, ma'am.
Okay. Good. Yeah, so now what we are going to discuss is about the protein folding and the processing in the ER. So what exactly happens in the ER? Okay. Yeah, sorry. So in the ER, so now if anyone asks you like ER ka function kya hai? So the first thing comes to your mind is that protein folding happens in the ER. Okay, and to be specific, in the rough ER. Okay, smooth ER ka function दूसरा है. If you know, it is meant for the lipid synthesis. We'll come to that later. So it is the site of protein folding, assembly of multi-subunit proteins, disulfide bond formation, N-linked glycosylation, addition of glycolipids anchors in some cases. Okay. Now again, please do remember, as these points are coming, I'll be telling in between itself. So N-linked glycosylation होता है आपका ER में, and O-linked glycosylation होता है आपका Golgi apparatus में. ठीक है? Do not get confused. Please do remember. And always disulfide bond formation is happening in the lumen of the ER. Correct? Now, what can you tell me by listening to this disulfide bond formation, ER lumen? What does it talk about the environment which is present in the lumen of the ER? What thoughts are coming to your mind when we talk about disulfide? What is it?
Oxidative environment. Disulfide bond.
Yeah, so disulfide bond formation means it will be oxidative or reduced.
Oxidative.
Sure?
Sure.
Okay. So this is how sometimes we do get confused. Okay. Now, in fact, the primary role of luminal ER protein is to assist the folding and assembly of the newly translocated polypeptides. अब polypeptides को उसके correct folding में लाना है, तो उसके लिए भी हमें help चाहिए, and that help is given by the chaperones. Okay, now we have talked about the chaperones, like there are certain HSP60, HSP70, Have you heard about these? What is HSP stand for?
Each of proteins.
Very good. So HSP 60 का दूसरा नाम क्या होगा? और HSP 70 का दूसरा नाम क्या होगा? Any idea? It's okay if you don't know because ये कोई पूछेगा नहीं, but still.
I don't know, but it's on the slide.
Yes, chaperones.
Sorry, what? I didn't get it. What?
HSPs are chaperones, right? So,
Yes,
I mean,
yes.
what are you expecting, ma'am?
No, I'm asking HSP 60, HSP 70, they are chaperones, but what else is their name?
Ma'am, on the slide it is BIP, but I didn't know this. It's just on the slide.
It's given in the slide, sorry. Yeah, sorry. So same way HSP 60 also, it has got different name and all. Okay. So in the case of HSP 60, it is known as chaperonins. Chaperonins. Chaperons, chaperonins are there. Okay. It's okay. So HSP 70, it is BIP. So it is thought to bind to the unfolded protein. See, here you need to understand that these are the chaperones which is helping in the protein folding of an unfolded polypeptide chain. Okay, then we do have the chaperones which helps in the folding of the misfolded proteins. Got the difference? We have got two types of proteins. एक होगा unfolded, जो नया नया ताजा ताजा ट्रांसलेट होकर आया है. एक होता है misfolded because of sometimes the pH difference or because of certain region it gets misfolded. Now, unfolded proteins, misfolded proteins, both needs guidance of the chaperones. So, in the case of unfolded proteins, these are your HSP 70, BIP, for example. In the case of the misfolded proteins, the other chaperones will be there, which helps in they try to bring the misfolded into correct folding. If it works, it's well and good. If it doesn't, then it goes for the degradation. Okay? So, one protein, what we have discussed now is the BIP, so which is present, which helps, as it crosses the membrane and then mediate the protein folding within the ER. अब इसका क्या मतलब हुआ? That the BIP proteins or the HSP 70, they are also known as the resident proteins. Now, what do you mean by resident proteins? Go with the normal English meaning. So resident proteins का मतलब क्या होगा? That they're always present in the ER lumen. ठीक है? So if suppose, now again I'm asking, if suppose your scientists or your interview panel people are asking, how can you locate in the cell alive the location, like till where the ER is spread inside the cell? With the help of the experiment or design an experiment and explain, तो आप कैसे बताओगे? Is my question clear?
No, ma'am.
Okay, I'll make it clear once again. I need space actually to write. So the question is, suppose as a interviewer, I'm asking you, design an experiment and tell me how in a live cell, okay, how can you show me the distribution of ER in the
We
cell?
can tag the resident proteins.
Correct, absolutely correct. So you're gonna tag the resident proteins, for example, BIP. So jahan pe aapko, when you say tag, always specify that you are gonna do the immunofluorescence. Okay, the technique what you're gonna do is the immunofluorescence. So this BIP protein ke against you're gonna use the antibodies, right? These antibodies will be tagged with certain fluorophore or chromophore, depending on the requirement. And then you will visualize under the microscope. So wherever you see the fluorescence, it means the ER is being spread till there inside the nucleus. Is it clear now?
Yes, ma'am.
Okay.
Yes.
Good. So that's how you have to build your, experimental designing thing. So now I'm sure everyone got, like, whenever you talk about resident proteins, it means they are the proteins always present in the ER. Okay. So now look it over here in this figure. So it is getting the huh.
I have a doubt. What is the difference between a chaperone and a chaperonin?
Okay, we'll come to that because an entire session is there, like chaperones and the chaperonins you're asking, right? We'll come to that.
Yes.
We'll come to that.
Okay.
Yeah. Okay. Anyways, both are the heat shock proteins only, but the difference, of course, it's there. We'll come to that. Another session is there for this only. Okay. So where was I? So again, as you see, the proteins, okay, so this is a part of the co-translation. You can see again the same thing. As soon as the nascent polypeptide chain is coming into the lumen of the ER, this bib protein comes and binds to it. Now together you can see so many bib proteins comes and binds and results in the proper folding of the ER. Okay, now here I would like to give one small assignment. See guys, when I say small assignment, it means it's your duty that you go back and study about it, because I won't be taking. It's a very simple concept and I don't want to spend time on that, and it is coming for the exam directly. उसमें कुछ ऐसे घुमा फिरा के नहीं है, direct question आता है, ठीक है? So now based on this proper folding of the protein, there is something called Leventhal paradox. So I want you guys to study about this. Okay. Leventhal paradox. Make a note of it, short notes. अगर आप पढ़ो नहीं समझ में आ रहा है, then definitely I'll take up this topic. But it's very direct and to understand like how, like what makes protein to be folded in a correct way. Like what determines, what dictates them. Like this is the correct folding. ऐसे ही fold होना है. यहीं पर ही interaction बनना है. यहीं पर ही disulfide bond बनना है. ठीक है? Like for example, in the case of insulin, let us say the structure is like this. It has got two disulfide bond, SA, or let us say that one is SA. But what determines that these are the correct disulfide bond formation? ये ऐसे भी तो बन सकता था, right? दो disulfide बनने के लिए 4 cysteine residues are needed. So why it is 1 2 3 4? Why not 1 3 2 4? तो ये कौन डिसाइड करता है? ठीक है? So these are what this answer will be given, understood if you know what is Leventhal paradox, and this is small assignment I'm giving you. Please go through it. नहीं समझ में आया, so let me know so that I can take up this topic. Okay. Yeah. So now this is clear, like how proteins are helping in the proper folding of the proteins. Okay. Now, correctly assembled proteins are released from the BIP, and then they will transport to the Golgi apparatus for the other modifications to happen. Now, if suppose abnormal folded is there and it is not getting properly folded, then they will be sent for the degradation. So whenever I say degradation, it is meant for the ubiquitin proteasome pathway. Okay. Now, the formation of your disulfide bonds. So between these disulfide bonds, you know that like how it is going to help in bringing the disulfide. So those cysteine residues hona bahut zaruri hai so that it will be brought together. Okay. Now in this ER, however, oxidizing environment is there which promotes the disulfide bond formation, and the disulfide bonds formed in the ER, which is playing an important role in the structure of the secreted and cell surface proteins. Now this sentence, it has got deep meaning. Okay. Disulfide bonds, which is formed in the ER, they play important role in the structure of the secreted and the cell surface proteins. Why? Why disulfide bond formation is so important in the secreted or the cell surface proteins? Can anyone relate? If it is internal proteins, there is not that much requirement of the disulfide bond formation. Whereas जो protein बाहर secrete out होता है, okay, I'll give you the hint. It is coming in exposure to the surrounding or the outside environment. And that's why having disulfide bond is good for them to maintain the structure. Now tell me, like, why it is said like this? Another hint I'll give you. The secreted proteins or the cell surface proteins are more stable structurally and functionally as compared to the internal proteins, which is present in the cytoplasm. Yes. Anyone? Tanvi, any idea?
No, ma'am.
No? Okay, no issue, no issue. See, when I'm saying that it is, when I'm saying that the proteins are more stable, okay, structurally and functionally, it is because of the presence of the disulfide bond formation. Now, disulfide bond is a type of a covalent bond. Do you agree? Right? So, covalent bond is the most stable bond as compared to the other ionic interactions or the salt bridges they form. So, in order to make your secreted protein more stable so that it does not lose its structure, so that it does not lose its function, they have got more number of disulfide bonds as compared to the proteins which are present inside the cell. Because outside, the conditions are not same as it is present inside. So, if they are secreted out, they need to face so many different environments. Okay, because for example, let us say insulin is there. Insulin is your peptide hormone, but it is a polypeptide, right? Now it has to travel a lot so that it gives the signal to the cells and so that they can, start taking up the glucose. ऐसा तो है नहीं कि वो अपने घर बैठ के आराम से सिग्नल दे रहा है. It is not. It has to travel a lot. During this travel, if suppose the conditions or infections or whatever it is, because of which it loses its structure. If it loses its structure, what will happen? There is no function, and hence this particular protein or hormone is not able to function, right? So I don't want that to happen, and that's why many of these secretory proteins or the enzymes are really very, very stable as compared to the internal enzymes. So more number of disulfide bond means the protein is more stable. Even if you heat the protein, okay, even if you heat the protein, we see that most of the proteins which has got higher number of disulfide bonds, they retain their structure. Now, once they retain their structure, they retain their function as well. Clear now? This point is clear?
Yes, ma'am.
Okay. So now we have got the another resident protein called disulfide.
I have a question. Like
Yes.
when you mentioned about the disulfide bond, the cytosol has a reducing environment, right? So don't they get dissociated when they enter into a cytosolic space?
No, so you need agents over there, right? See, with the help, making a bond, breaking a bond, it's all about delta G. Correct? You need energy.
Okay.
Right? You need energy to make a bond, to break a bond. Right? And this energy, see, this is what the enzymes are doing. अगर आप उसको उसी कंडीशन में छोड़ दोगे, so it will take time to break itself, it will take time to make a bond. What enzymes are doing? They are just reducing that gap so that they can easily form the bond. Now, making a bond means what? Here the PDI or the protein, whatever it is, protein disulfide isomerase, it is helping because it has got its own disulfide. अब ये क्या करता है? The extra H group जो यहाँ पे present है वो खुद ले लेता है. So that the disulfide bond is formed. Now even this PDI, okay, it has to lose this H so that it can form back its own SS bond. Getting the point? Now, once it is formed and these are released where you have got the reduced, where the chances of SS to become SH is more. But breaking this bond needs again the enzyme so that it can get reduced. जब हम lab में करते हैं, we are using the catalyst to carry out such reactions, right? In the same way, here enzymes are helping in carrying out such reactions. But even though if you have got the reduced environment, but you don't have catalyst to break the bond so that they can get reduced to SH, then it is not possible. Did you get my point, Sanil?
Yes, yes.
Yeah, okay. It's always about delta G, okay? Always remember about the delta G. Okay. So the another resident protein in the ER is your PDI. Okay, so first one is your BIP protein, the second one is your PDI. Now, proteins which are glycosylated on specific asparagine residues. So as I said, ER, one second. Okay, so as I said, at the ER, you have got the N-linked glycosylation, and N is only possible in the case of the asparagine. Okay. Now, oligosaccharides, around 14 sugar residues, they are added to the asparagine residues of a growing polypeptide chain as they are translocated into the ER. Now, these oligosaccharides, usko पकड़ के रखने वाला कौन है अपना lipid carrier. Okay. Now, then it is transferred as a unit to acceptor asparagine residues in the constant sequence of asparagine X serine threonine. So, ये आपको again, this is something which again you need to remember. Any protein which has got this sequence of N, X means any amino acid followed by serine or threonine. So, to this your oligosaccharide will be transferred. Okay. So, right now what we are saying is that the proteins are getting, they are getting the glycosylated. So does it mean that each protein is getting glycosylated? Whatever protein gets translocated into the ER lumen, does it mean like that?
No, the one which would have a signal would get the glycosylation.
Correct, absolutely correct. So it is not that all the proteins are getting glycosylated, only the glycoproteins which forms, and having this sequence only they will get the glycosylation. Okay, good. So this is how it looks. So here again, slowly we are adding all those, lipid carrier and those proteins name into that, and you need to remember all this. So you have got the protein, and see, we saw that there is an asparagine which is supposed to, all this oligosaccharide needs to be added to that. So the lipid carrier over here is known as dolicol. Okay, now to the dolicol, the phosphate group is there, to which this 14 sugar oligosaccharide is present. Now this 14 sugar oligosaccharide is fixed. Your dolicol is fixed. Now, depending on the requirement of the glycoprotein formation, the number of glucose or mannose or N-acetylglucosamine, they are going to change. Okay. The first step which happens in the glycosylation is that from the dolicol, this portion, okay, from here, it will get transferred to your asparagine. So what do we see? We have got 2 molecules of N-acetylglucosamine, okay, 9 molecules of mannose and 3 molecules of glucose. This is something which you need to remember, okay. Again, I repeat, 2 molecules of NAG, that is N-acetylglucosamine, 9 of mannose and 3 of glucose. Okay, so this entire oligosaccharide is gonna transfer from the dolicol lipid carrier to your protein, which is having the asparagine. Okay, now once this is transferred, one by one the glucose molecules are removed. After this, having the 2s2 NAG and 9 mannose, depending on the requirement कि ये protein आपका किस तरह का glycoprotein बनना चाहिए in future, all those processing will take place. The addition and deletion will be happening, but this much is happening in the ER lumen. Okay, all good?
Ma'am, is this specific oligosaccharide for mammals, or is it like every single eukaryote may this oligosaccharide is the one that's added?
This is general. This is general. Okay, so this is happening in the eukaryotes. Then when I said the required or addition deletion will happen, that is depending on the function of the protein. Okay, because the dolicol is carrying these sugars only. अब इसके ऊपर you add or you delete. So again, some proteins, they are attached to the plasma membrane by the glycolipids rather than by the membrane spanning regions of the polypeptide chain. Okay. The another example is of your GPI, that is your glycosyl phosphatidyl inositol. This also we have discussed during our plasma membrane lectures. So these anchors are assembled in the ER membrane. They are then added immediately after the completion of protein synthesis to the C terminus of the proteins. Okay. Here, look at over here. So where your GPI anchors are there, right? So these proteins are, the proteins can be anchored to the outer face of the plasma membrane by the by the glycolipids attached to their C terminus. Are you getting it? तो यहाँ पे क्या हो रहा है? This is your GPI. प्रोटीन आपका यहाँ पे है, वहां पे क्या था, बीच में कहीं पे आपका एस्पेराजिन आता है, टू विच द ओलिगोसैकेराइड हैज बीन एडेड. In the case of the GPI, you have got this glycosyl phosphatidyl inositol ready. Okay, to this the ethanolamine present group is present, it goes and makes a bond. So if you see, this is CONH bond. What is CONH bond? It's a peptide bond, correct? So it forms with the C terminal of your protein. Now this entire thing becomes your GPI anchor. Okay, in this also, please do read this thing, that, and you have to remember also, this is the GPI anchors are joined with the C terminal, and this GPI is made up of 3 mannose and 1 glucosamine residues. Okay, then again it goes and binds to the inositol head of your phosphatidyl inositol, and that's how it becomes a GPI. Okay, so this is something you need to remember. Now, why I'm asking you to remember is they're not going to ask you directly कि GPI में क्या क्या होता है या end-linked glycosylation में क्या होता है. The question will come when we talk about the enzymes involved in the processing or the formation of the GPI or the other groups, which will come later eventually. Okay, so those enzymes are acting at which point of your ER protein folding. That time it is important to remember the previous ones. Okay. It's simple, don't worry about it. Now the addition of the GPI anchors, same thing. So they are assembled in the ER and added to the polypeptides anchored in the membrane by a C-terminal membrane spanning region. So you can see it over here. ये आपका protein है यहाँ पे आपका oligosaccharide है, fatty acid से attach है यहाँ पे आपका, right? Now this group, ethanol amine group, will make your what to say, the peptide bond with the C terminal of your protein. So ये यहाँ पे attach हो जाएगा. Now whatever it is remaining, जो इतना आपका hydrophobic region आपका remaining है, what happens to this? It is no longer a functional protein, so it will go for the degradation. Guys, are you able to follow me?
Yes, ma'am.
Okay. Yeah. So that's all, guys, for today. This much I was able to prepare. I'm just stopping here now.